Okay, domain 4 is reproducibility of results in a range of clinically relevant conditions, otherwise known as external validity. What's the purpose? The purpose is to assess whether the results were reproduced in a range of clinically relevant conditions. And what do we mean by range of clinically relevant conditions? This could include different methods of disease induction, different genetic composition, different ages, different sex. Now, the underlying rationale behind preclinical animal research is the genetic, anatomical, physiological, and biochemical similarities, which could be one or more of the above, between animals and humans. Different animals have different levels of genetic similarities with humans and between each other. This leads to anatomical, physiological, and biochemical differences between different species, which of course may result in differences in the treatment effects between different animal species or different models of induction of disease in the same species. The differences may be in the direction. What do we mean by that? That means the intervention is beneficial in some species and harmful in others, or in the magnitude. That means the intervention is beneficial in all species, but the treatment effects differ in different species. For example, in one species, it might be 50% effective, in another, it might be 25% effective. Now, even if the inconsistency is only in the magnitude of effect, this indicates that the treatment effects in humans may also be different from those observed in different species. For example, if the treatment was 50% effective in species, let's say mice, and 25% effective in rabbits, it may be only 1% effective in humans. So, consistent treatment effects observed across different animal species or different models of induction of disease may increase the likelihood of similar treatment effects being observed in humans. Now, how will you classify this particular question? Classify this as yes or probably yes if the results were consistent across different preclinical models, and that could be part of the study itself. So they could have done some observations in mice, and they could have done some observations in rabbits, or it could be done as a part of different research by the same team, so they cite their work, or it could be somebody else's work. And you're looking at whether the intervention was consistently better across the studies and the effect was large in all the studies. Of course, if the intervention was worse than control in all the studies, that is also consistency. And if the differences were small in all the studies, then that's also consistency. Classify this as no information if you don't know whether the research was performed in different preclinical models. So if you don't know whether they used different preclinical models, then you would classify it as no information. And if you don't know if the results were consistent across different preclinical models, then again you would do it as no information. Now, classify this as probably no if it was clear that the research was not performed in different preclinical models. So here the authors say this is the first or experiment of this type in any preclinical model. Okay, so it is clear that the research was not performed in a different preclinical model. Or the authors say the results were clearly inconsistent across different preclinical models. They say, okay, we did this in mice. In our previous experiment in rabbits, we got different results. Okay, so either if the research, if it was clear that the research was not performed in different preclinical models, or if it was performed and it was clearly inconsistent, then you would classify this as probably no. Signaling question 4.2, where the results consistent across a range of clinically relevant variations in the model. In the clinical setting, the treatment is used in people of different ages, sex, genetic composition, and with associated comorbidities. Now, these differences within the species and existing comorbidities can lead to different treatment effects even if the same species and the model of induction is used. And what we want to know is whether they have used a range of genders, different age, comorbidities, etc. So classify this as yes or probably yes if animals of multiple ages, sex, genetic compositions, and existing comorbidities were used, and if the treatment effect was consistent across these clinically relevant variations. And here, of course, the same thing goes. If the intervention was consistently better than control in all the studies, and the differences were large in all the variations. Classify this as no information if it was not clear whether animals of multiple ages, sex, genetic compositions, and existing comorbidities were used, or if the treatment effect was consistent across the clinically relevant variations. Okay, so one situation is we are not clear whether there was clinically relevant variations. Another situation is there were clinically relevant variations, but the authors did not report whether the treatment effect was consistent across these variations. Classify this as probably no if the authors used animals of similar age, sex, genetic composition. For example, they might define we used mice, specific type of mice from a specific litter, for example. That means the genetic composition is more likely to be similar, and also only one gender. And they didn't mention about comorbidities. They actually say they did not have any comorbidities. So classify this as probably no if the authors used animals of similar age, sex, genetic composition, or if it was clear that the results were not consistent across clinically relevant variations. So they actually used a lot of clinically relevant variations, but the results were not consistent. Signaling question 4.3, did the authors report after taking existing evidence into account when choosing the comparators? Okay, so the researchers may choose an inactive control rather than an established active treatment as the control to show that a drug is effective. They may also choose a weak control, such as a dose lower than the effective dose or an inappropriate route for control, to demonstrate the benefit of the intervention. Now, experimental results that the intervention is better than control are applicable only for the comparison of the intervention with a weak control, which may not be clinically relevant. So how do we classify this? Classify this as yes or probably yes if the authors chose an established active treatment at the correct dose or route, in case of pharmacological interventions, as control with evidence. So they actually say this is what we chose as control because this is the current treatment provided for this disease. So that's why we chose them as control. This is the correct dose and route by which this is actually administered. Classify this as no information if the rationale for choosing the control or the dose or route for the control was not reported. Classify this as no or probably no if the authors clearly stated the established active treatment but chose an inactive control or used the inactive treatment at the incorrect dose or route. So they clearly stated currently the standard treatment is oral aspirin at 75 milligram, but we actually chose a different different control We chose no control, for example. That's problematic. We chose aspirin dose, which is equivalent to 10 milligram in the humans, um, or we chose to give it intravenously. So all of these are problematic. So classify this as no or probably no if the authors clearly stated the established active treatment but chose an inactive control, or use the active treatment as control at the incorrect dose or route.